undergraduate degree from Yale in physics, and then she moved to Harvard to do her PhD in the Psi Lab. She's finishing up, so she's going to graduate hopefully this May, I think is her plan. I met Elizabeth at the Gordon Conference last summer on microbial population, genomics, genetics, who knows? And she was one of the selected student speakers for the conference, and at least to my mind, she was by far and away the best student talk of the conference, and certainly one of the best talks of the entire conference, including all of the invited speakers. So when Elizabeth said she was going to be coming out to Stanford, it was a great opportunity for her to present her story to our larger CEHG community. So Elizabeth, welcome.
Thanks a lot, Gavin. So thank you all for coming today. As Gavin said, I'm a PhD student in Michael Desai's lab at Harvard, where I study theoretical evolutionary dynamics and experimental evolution. So as many of you probably already know, experimental evolution is based on the following extremely simple experiment. So you take a microbe of choice and grow it in, say, a tube of medium, and at the end of the day, take a small sample from this population and transfer it to some fresh medium. Repeat this process over the course of months to years in the lab as these populations undergo hundreds or thousands of generations of evolution. So perhaps the first surprise from this kind of experiment is that basically regardless of the exact conditions that you choose, these populations will reliably adapt. That is, their competitive fitness, which is roughly equivalent to their growth rate relative to their ancestor, increases over time. So now that we have modern sequencing technologies, we can actually look under the hood, so to speak, at the competition among lineages that's driving this adaptation. So, you know, the reason that these populations increase in fitness is that sometimes individuals will get a mutation that allows them to grow faster, and they will preferentially outcompete their fellows. And we can actually now look directly at this process. So this is, for example, a population of bunny yeast that from an experiment from our group. Where each line here is the frequency of a particular de novo mutation that arose over time. And what you can see is that there's one lineage that carries, you know, a collection of mutations, at least one of which had to have been beneficial, that drove it to high frequency in the population, but then it was later outcompeted by this other even fitter lineage. So the kinds of things that we would like to maybe be able to do now that we can directly measure these kinds of dynamics is first to predict things about the evolutionary process, like, for example, given a mutation that confers a particular growth advantage, what are the chances that that mutation will take over the population? Maybe outcomes like how fast will populations like this adapt? And then we can also probe things like how these kinds of outcomes depend on the evolution condition, on the genotype, etc. And of course, it's hard to learn things about the underlying evolutionary process just from one of these populations, because evolution is a stochastic process. You know, exactly which mutations arose in this population was random, when they arose, etc. But luckily, with microbes, it's straightforward to evolve many identical replicate populations and measure distributions of outcomes. So for example, say I evolved 20 populations in environment A, and they might have this distribution of final fitnesses in some other environment B. And from that, we can learn things about the underlying parameters in the evolutionary process, like the available beneficial mutations. So, and another nice thing that this kind of parallelism lets us do is identify genes that might be selected. So, for example, these are you know, genes that perhaps had more mutations in them in independent replicate populations than we would have expected, and we can identify these as targets of selection. So today I'm going to use this sort of general strategy to address two different questions about evolution. One has to do with evolution in different environmental conditions, and the other with the influence of starting genotype, the sort of starting location in the fitness landscape, on adaptation. So as we all know, specialization is quite common in the natural world, and a long-standing question in evolutionary biology has been whether or not when we see specialist organisms, this is because of sort of fundamental engineering challenges, that is, it's just physiologically difficult to be an organism that's good at growing in many environments, or whether it's more a byproduct of the evolutionary process, that is, these generalist organisms often exist, but evolution just tends not to take that path. And a way to state this at the molecular level is that we'd like to know whether when a population adapts to a particular condition, the driver mutations that allow that population to adapt are the same ones that incur some kind of cost in another condition, so this is antagonistic polytropy, or when we see trade-offs in other conditions, they're just more a result of the buildup of passive adaptation. A slightly different statement of this question that's sort of more relevant for the dynamics of evolution in changing environments is whether or not when you have a population adapting, the mutations that it tends to fix are biased to be better or worse than the typical mutations in another environment. So I chose to address this question in the context of populations of yeast adapting to different sort of canonically stressful conditions. So I founded 260 populations from the same clonal progenitor and evolved 20 populations, so populations in each of 13 different conditions. So these conditions are all based around a standard yeast laboratory media, some kind of complete with 2% glucose and at 30 degrees Celsius, sort of standard yeast lab conditions, unless otherwise noted, and have other kind of stresses added. So for example, additional salt, a different fixed pH, higher temperature, etc. So I evolved these populations in batch culture for 800 generations, and at the end of the day, picked one clone per population for further analysis. So the first thing that I was interested in knowing is whether or not these populations, have gained and lost fitness in different conditions. So I did sort of standard competitive fitness assays, mixing fluorescently labeled version of the ancestor with each evolved line, and measuring the relative frequency of these two strains in competition over the course of a couple of days. And the change in these relative frequencies over time is a measure of their fitness. So for example, if we do this for one clone adapted to one of these conditions, we can see, in this case it evolved in high temperature, it's gained a little fitness in high temperature, and this is its fitness across a series of other conditions. So looking at all of the clones in the experiment, now each panel is the 20 clones that adapted to a particular condition. And what seems to jump out here is that there are certain environments where it seems like sets of clones from all of these different conditions have lost fitness. So this is this low temperature environment, lactose, and actually the normal 30 degrees Celsius environment. We can pull this out a little bit more clearly just with unsupervised hierarchical clustering. And so you can see that there's somewhat similar looking fitness losses that occurred sort of in different proportions in these different environmental conditions. Looking more closely at one of these sets of clones, in this case adapted in high salt, so they gain fitness in high salt, as one would expect. About half of these lines experienced a significant trade-off in these three environments, and about half of them did not.
The 30-degree environment seems weird. They were losing fitness in their own home environment.
Yeah, I'll come back to that at the end. Thanks. Yeah, that's true. That's true. So again, going back to these original questions, we're interested in knowing whether or not these trade-offs that we observe are caused by the same mutations that help these populations adapt, for example, to high salt, or whether they're generally caused by other passenger mutations. So of course, to answer this question, we need to know which mutations cause the trade-offs. First, I did whole genome sequencing on all of these endpoint clones, called about 10 mutations per clone. Generally, they're spread throughout the genome, but there are a number of targets of selection. So, for example, if we look at all of the genes that were mutated in 3 or more independent lines in this experiment, you can see that there's quite a number of putatively selected genes, and this is only a subset of the ones that are probably selected in this case. And some of them appear to be specifically beneficial in particular environments. So, for example, the silent information regulator family here was mutated mostly in the high temperature and ethanol conditions, whereas some of them don't have any particular statistical association with any condition. So this is 2 of our favorite experimental evolution genes in yeast. These come up in these kinds of experiments all the time. So we have a number. So we have to see a number of mutations that are selected, and the question then is, okay, so do any of these mutations cause this defect? So the simplest possible model that we can imagine is that there's some common cause of the defect. There's some one mutation or perhaps one complex where when you mutate it, you get these sort of parallel effects. So this is an easy hypothesis to test. We can just look at the mutations that we call. So, for example, again, looking at these lines that adapted to high salt, and now organizing, so sorry, I should have mentioned this earlier, in these kinds of tables, each row here is a particular clone, and each column is a gene, and a black bar just indicates that that clone got a mutation in that gene. So organizing this table based on whether or not these clones have a fitness defect or don't have a fitness defect, we immediately see that there's no one gene here that can account for this fitness defect. There's nothing here where if you mutate it, then you have the defect, and if not, then not. So there's no simple sort of common cause, but it's still possible that these fitness defects are caused by a single mutation, but maybe it's a different mutation in every line. So to address this possibility, I did 2 types of experiments. One is that we can reconstruct particular mutations that occur during evolution on the ancestral background to test them individually. And the other is that we can produce progeny that have subsamples of the mutations in the evolved lines in classical genetic sense by back-crossing to the ancestor. So, for example, in this first type of experiment, these are the 6 non-synonymous mutations that occurred in a particular line that was adapted to high salt. This line is shown in black here, and what you can see is that 2 of these mutations are beneficial, and they together account for the fitness increase that we saw in high salt, but absolutely none of them have this fitness defect. So it's possible that the fitness defect is still caused by some mutation, but just not one of the ones I reconstructed, maybe an intergenic mutation, maybe even a mutation that I didn't call in sequencing. So to address that, I back-crossed this and a number of other clones to the ancestor to look at, again, these subsamples of the mutations. And when I do that, I see something a little bit odd. So as a control, you can cross this line to itself, just to check that the fitness defect survives meiosis. But when back-crossed to the ancestor, you see that the defect is an all or nothing effect. But it only appears in a few of the progeny. And that implies that these guys were the unlucky ones that got a particular combination of mutations that caused this defect. That is, there's some kind of higher order epistasis. Either that, or the defect isn't caused by a genomic mutation at all. So we're left with 2 seemingly somewhat unlikely hypotheses. Either there are, different combinations of mutations, and it has to actually, based on the sequencing results, it has to be a different set of mutations practically in every single line that has this defect that causes the defect. So that's a huge amount of higher order epistasis. Or alternatively, it's caused by something that is not in the nuclear genome. All right. So in the course of doing all these experiments with these lines, I noticed something slightly odd about them, and that was that although they always lose to the ancestor in competition, they seem to actually grow fine on their own. And these sort of magnitude of fitness defects that I'm observing here should actually be readily apparent when you grow them in a tube or just on its own. And so that implies that there might actually be some kind of direct interaction going on between the ancestor and the evolved lines. So to look at this in more detail, I checked to see whether the competitive fitness that we measure was dependent on the starting frequency of the two lines in competition. So this is, again, this 21C environment where we see these fitness defects. And so it turns out that the initial frequency of these two competitors strongly determines how big this disadvantage is. So the more of the ancestor that was present in the competition, the worse the evolved line does. And so this directly implies that there's some kind of either inhibition or maybe even killing going on between the ancestor and the evolved line. So one potential mechanism for this is the yeast killer virus system. Yeast usually have a virus that floats around in its own little capsid in the cytoplasm, and sometimes there's also a little satellite virus, so I should say that this is a double-stranded RNA virus. Sometimes there's also a satellite virus that encodes both a toxin that's secreted from the cell and an antitoxin that protects this cell from the toxin it's secreting. So the hypothesis here is that my ancestor might have carried one of these satellite killer viruses, and in the course of evolution, some of these evolved lines lost it. And so then the evolved line, the ancestor, would actually literally be killing its descendants. So to test for this, I extracted genomic DNA and double-stranded RNA from the ancestor and from a collection of lines with the fitness defect and a collection of lines without the fitness defect and ran them on a gel. So I should mention that this main virus here is about 4.6 kb in length, and the satellite virus is about 2 kb. So you can see that this is actually true. My ancestor has this 2 kb killer virus. All of the evolved lines with the defect have lost it, and the evolved lines with no defect have retained it.
Does that mean that the killing is very inefficient or absent in the presence of high salt?
Yes, so I'll show you data on that in a second. Yeah. So so the short answer is it's affected by things like temperature, pH, salt, somewhat, but not entirely. So this is a strong correlation, but to establish causation, I took the ancestor and cured it of the virus, and then recomputed it against these evolved lines with the defect, and the defect should be alleviated if this is the cause, and that is indeed what we see. So it looks like this sort of major feature that we saw in the data, these parallel types of fitness losses in different environments were actually caused by the loss of this yeast killer virus. This might be a good time to go back to Dimitri's question. So because this defect is non-transitive, it can fix in these lines, even in their own environment, because the competitors that were there at that time maybe we're not killing.
Do you know if the loss is adaptive or is it just a drift mechanism?
Yes. So Gavin's question is exactly the next one that we wanted to ask, which is whether, so how did this evolve, right? Was it actually selected in particular environments, or did it hitchhike? So to address this, one can just compete the ancestor that has been cured of the virus against the ancestor that still has the virus to just measure the effect of losing it in these different conditions. So this is, again, this fitness as a function of frequency. Again, losing the virus has this large frequency-dependent effect in the low temperature condition. And it actually turns out to be deleterious in all of these other conditions as well. Although, interestingly, it's the least deleterious in this high temperature condition where it was lost the most. So this implies that the virus loss had to hitchhike on the sort of rapid adaptation that was occurring in the population to fix. So there's an alternative, slightly more complicated model for Dimitri's question, which is that it's actually also possible that for the virus to lose killing ability, but before it is itself lost. So it's possible that this hitchhike to high frequency first, and then virus loss could occur. But it's certainly true that this was not selected. It fixed as a result of this sort of evolution that was going on in these populations. So finally, now that we know about this virus, we can isolate the effects of just the genomic mutations by competing evolved lines against the cured reference. And what we see here is that the genomic mutations actually tend to provide benefits even in environments where these clones were not evolved. So these are groups of clones that were evolved in these environments here, all measured in high salt. And you can see the ones evolved in high salt adapted the most. But most of these other groups of clones also tended to have increased in fitness in high salt, with the exception of this group evolved at high temperature. And I actually am showing this particular environment because this was the only case where a group of clones evolved in one condition on average lost fitness in another condition. So we can also see this at the level of individual mutations. So for example, these are mutations that in blue that arose in high salt clones and in red that arose in high temperature clones. And you can see that either, in this case, these adaptive mutations don't give any trade-off in other conditions. It's measurable, or in this case, there's maybe a weak measurable trade-off, but not very much. So in conclusion, the trade-offs that we saw that arose during adaptation were mostly caused by the loss of the yeast killer virus, and this is sort of it's a non-transitive competitive effect. This change itself was deleterious, which implies that it had to hitchhike with other beneficial mutations, so that it really was a product of sort of this evolutionary process. On the other hand, the adaptive nuclear genetic mutations show either weak or no trade-offs. And we can tell from looking at what happened to these clones over the course of evolution that the mutations that they fix tend to be biased to be better than average in all of these other conditions. Yeah?
Can I?
Yes.
So I'm not really sure I understand the hitchhiking hypothesis.
Yeah.
So the hitchhiker is better than the ancestor in the presence of the virus. The total fitness has to be higher.
Yes.
But then eventually it ends up being lower.
Well, so it's lower. So I see what you mean. So yeah. So I guess that would strongly argue for, so I guess I would strongly argue for either some other more complicated epistasis or this two-stage process that I was suggesting, which I actually have other evidence is probably what happened also.
Is the loss of the virus, does that occur at a very high rate?
So that's
usually these are done in really high population crops so that you don't have drift coming in.
right. So yeah, that's another really good question. So the virus itself is not lost at a high rate, but this either mutation or epigenetic switch to the state where the virus, where the swine is still resistant, but it doesn't kill anymore, does happen at a high rate. So that's the other reason that I think that most likely what happened was actually this fixed first, and then additionally the virus loss fixed. Yeah. All right. But I mean, still, even this sort of neutral change of the loss of killing would still have to hitchhike to get to high frequency in the population. All right. So yeah.
I'm just wondering, could the existence of the virus at the 30C and the fact that that virus doesn't seem to work very well in almost any other condition, isn't that an engineering constraint?
So I guess I would argue that these fitness effects, so 30C is actually this blue here. It's the most effective at the low temperature. And I guess I would argue that under, these are actually, fitness effects of a few percent are actually pretty large. So that should be enough under many scenarios to retain the virus. That is, it works well enough. But yeah, I mean, in the presence of these other kinds of mutations that you can get that aggregate killing and so forth, not totally clear.
Sorry, one more question.
Yeah.
So even though your global environments are different, they're also similar.
Yes.
In that they're a robot, same volume.
Yeah, absolutely.
So could it just be that?
That's why we see correlated adaptation other than, so this is definitely possible. I guess the only other thing that I would say there is that at least the measurements that I've seen of, for example, the yeast deletion collection measured in different environmental conditions where they, in this case, typically varied the carbon source or added a drug or something, and sometimes they tried environments that were as different as like oak, for example, to try to simulate something that yeast lives in. And in those cases also, if you look at if you try to draw sort of the joint distribution of fitness effects of these mutations in a couple of environments, there are some that are uncorrelated, but you basically never see a negative correlation. I have yet to see a negative correlation. So this is obviously a slightly different statement from the ones here, but this sort of, to me at least, implies that there are a lot of these mutations out there that are beneficial in both conditions, and perhaps, I have yet to see a condition where the beneficial mutations are biased to be worse in another one.
Why didn't they fix them?
Why didn't they fix that? So that's a good question that I've also thought about. And so yeah, I mean, these experiments, so I guess there's a couple answers to that. One is, these experiments also have this limitation that they are all lab conditions and they're all basically measuring growth rate, right? So, there's potentially many other types of things you need to do to survive in the wild that aren't being assayed here. I guess another thing is that it is possible that, if you go to enough different environments, many of these mutations will be deleterious somewhere. But yeah, we can talk more about it later if you want. I'd be happy to. Yeah.
So can your growth curves tell you whether the selection coefficients of these mutations that are beneficial everywhere are somehow amplified in the stressful environments?
I'm not quite sure I understand the question.
Like if you were competing like the mutant against an ancestor, like if the mutant goes to fixation in all the environments, does it go faster in the stressful ones?
Right. Yeah, that's a great question. That one could. So I think it's likely to be correct that, for example, the fitness effects just tend to be of larger magnitude in, for example, the high salt condition. And yeah, I could definitely look for that systematically, and that would be interesting. Thanks. Any other? All right. So great. So next I like to tell a different story about how the starting genotype of a population influences the adaptive process. And specifically, we're interested in how starting genotype influences both the rate of adaptation and the mutations that fix during adaptation. So to address this question, we decided to take advantage of a nice resource that was made in the Kuliak's group. So what they did was to cross a lab and a vineyard strain of yeast. This is BY and RM for yeast people. And they picked a bunch of segregants that each differ from one another just based on the sequence similarity of these guys at about 30,000 loci. So what I did was to subsample 230 of these genotypes and found 8 total populations for each genotype, 4 of which will evolve in sort of the normal 30 degrees Celsius condition and 4 at a higher 37 degrees Celsius temperature. So just to be clear, each of these populations starts out clonal for a particular one of these genotypes. So I evolved it in the lab for 500 generations, during which time mutations can occur that may depend on the genotype or on the environment. And the nice thing about the structure of this experiment is that if there are sort of differences in the adaptive process that depend on genotype, because each locus that's present in one of these strains is also in half of the pool of founding genotypes, approximately, we can potentially map these differences back to individual loci and founders. So again, the first thing that we were interested in is how genotype influences the rate of adaptation. So again, I did these competitive fitness assays where now I'm going to be measuring the final fitness of these populations that are adapted minus the initial fitness of this founding genotype in this environment to get the fitness, the fitness gained. So first we can look to see whether there's systematic variation at all among the genotypes in how fast they adapted. So this is, for example, the 30 degrees Celsius environment, and each dot here is the average amount of fitness gained by the 4 populations that descended from a particular genotype. And the bars are standard errors about those means. And the genotypes are just arrayed in a random order here. So you can see that even though, of course, evolution again is somewhat random, what happened to each of those 4 populations was different, but yet there is still systematic differences in the average rates of adaptation that depend on the genotype. So the classic way to now try to explain this kind of variation is to build a linear model of the fitness gains from individual loci in the founders. And of course, we have to include only a subset of those loci in this model because otherwise we'd be way overfitting. So I did sort of a standard iterative procedure to discover QTLs, as it were, and decide which loci to put in here. So we can see that this predictor, which I'm calling the trait value, which is a linear combination of these special loci, does kind of an okay job at explaining some of this variation that we saw in the rate of adaptation, where this is, again, this 30 Celsius environment, and this is the high temperature environment. Okay, so an alternative method of trying to predict the fitness gains was suggested by some earlier work from our group, and also a lot of work that's been done by other groups, that has shown that the amount of fitness gained is strongly dependent on the initial fitness of a population. But both in the case of this experiment and in these others, these starting genotypes have only differed from one another by 1 to a handful of mutations. So it's quite possible that starting from much more different genotypes that are much further apart in genotype space. There are now many more loci for the new mutations coming in to interact with, that this clean pattern would wash out. But at any rate, we can give it a try. And it turns out that actually initial fitness is much better at explaining the fitness gains that we see in both of these environments than this linear model with individual loci, even though these models have 7 parameters and these only have 2. And so this is kind of an interesting example of how, in this case, fitness is some nonlinear function of the underlying genotype, presumably. And that if you sort of know, in this case, we had sort of a lucky guess of what the correct sort of nonlinear function is, that can actually do a lot better sometimes at capturing a trait than these linear models that we can build. So this is a nice dependence to see, but it raises the specter that maybe fitness is the only informative part of the genotype. Maybe we're not going to get any additional information at all from all of these individual loci. So one can see whether or not one gets whether or not we can improve our predictions by adding any particular loci to these models that have initial fitness. And there's only one locus actually that helps, and that is this one pictured right here. So you can see that the identity of the allele at this locus strongly affects the initial fitness in both environments. But it also affects the rate of adaptation at a modest level over and above initial fitness in this high temperature condition. So for example, if you have this intermediate fitness and you're green, you systematically adapt slower than if you were yellow. All right, so we've seen that the main dependence of the rate of adaptation on genotype is through fitness, but with this small additional effect of this particular locus. So now we'd like to know whether or not the mutations that occurred during adaptation, sort of the paths, if you will, that the populations took, also depend on genotype in any way. So to address this, I did just mixed whole population sequencing of about 250 of the endpoint populations, again shown as rows here in this table. And these are all of the genes that were mutated in 2 or more of these independent populations. And we can get a little more information from this by organizing these into pathways. So it turns out that there's 87 genes here involved in ribosome biogenesis. And actually these are all in a large complex that builds the small subunit of the ribosome. And there are also a number of mutations in our favorite pathway, the RAS pathway. And interestingly, these mutations tend to be loss of function mutations, so premature stop codon, small indels, whereas these mutations are all putatively gain of function. They're single nucleotide polymorphisms in essential genes. So you might have noticed that one of the genes in this locus that I said influenced the rate of adaptation was actually this gene here, pre-3. So one might wonder whether or not the mutations in this gene and all these associated genes have anything to do with which allele state you have at that locus. I was also going to mention that we can think of these as a module, sort of also in the sense that even though we sometimes do see 2 mutations in these genes in the same population, these are depleted relative to what we would expect by chance. All right, back to whether or not are these genes associated with this locus. So it turns out that they are, that all 87 of these mutations in these genes are in individuals that started with the deleterious allele at this locus. So these are all putatively compensatory mutations. And one just so story about why they occurred is maybe that, so the deleterious allele is carried by the vineyard strain, and it's possible that that the vineyard strain living at more temperate temperatures was able to tolerate some kind of mutation that destabilizes this complex at higher lab temperatures, and that all of these mutations restabilize the complex during evolution. So finally, we'd like to know, we see that we can get some information about which mutations a strain is going to get based on whether or not it has this one particular locus. That's very clear from the other table. And so we'd like to know whether or not the other loci that maybe affect initial fitness also have any predictive power to tell us about which mutations the populations are going to get. And unfortunately, the answer is no. So we can address this by measuring the mutual information between knowing the allele state of at a particular locus and which mutations are going to occur. So you can clearly see that this, as we already knew, knowing this allele state at this 333 locus gives you information about the mutations. But once you've already controlled for the 333 locus and the evolution condition, knowing about any of these other additional loci doesn't give you any more information about the mutations. All right, so finally, to try to answer these questions, the rate of adaptation depends on the genotype in this experiment, primarily through the fitness of that genotype, with a small additional contribution due to this particular locus that we found. This initial fitness is a better predictor of how much fitness these populations are going to gain than a sort of standard linear model of QTLs. The reason that this statement about initial fitness being affecting the rate of adaptation is sort of interesting to us is primarily because it implies that there's some regularity in the distribution of fitness effects available to evolving populations as a function of their fitness. That is that maybe it's not different in any different genotype, but it primarily depends on the genotype's initial fitness. And finally, in terms of the mutational paths that these populations took, they tended to be similar, with a major exception of this one P33 locus that opened up a whole new mutational path that these populations tended to explore. And with that, I would be happy to take additional questions and thank my whole lab, particularly my advisor, Michael, and Sergei Kuzhinsky, who is currently starting his own lab at UCSD and was my direct mentor on both of these projects.
So the mutational target and the ribosome biogenesis genes,
Yeah.
does it explain, and the fitnesses and the size of the target, does it explain the extra rate of adaptation that these clones experience?
So the weird thing is that for their fitness, they adapt slower, right? So let me go back a few slides. All right, so if you have the deleterious allele, you actually, you say, in the only place where I really have guys that have the same fitness, you actually adapt slower if you have the deleterious allele than if you have the beneficial one. So the way that, one way to think about this, and we don't know if this is the right way, but is that there may be other phenotypic traits that need to be optimized to grow well in these conditions. And that basically, if you think of this locus as being sort of outside of this, the main sort of diminishing returns pattern, then these guys on the end have sort of all of the other right phenotypes, except that they have this one bad locus, right? So they have sort of fewer of the other things left to optimize. And so in some sense that they're more like these populations over here that have the better allele at this one locus, but also are on the good end in terms of all these other phenotypes. We don't know if that's true, but that's sort of one way to explain what's going on here.
So if you were to compare the adaptability of the different strains between 30 and 37, say conditioning on their fitness,
Yeah.
so that's no longer a factor, maybe also in the genotype at Area 33. Have you seen, is there any other correlation between them that would imply some kind of perhaps other contribution of genotype maybe that you can't detect from the single locus?
Yeah, so that's a really good question that I am actually planning to look at in more detail and have not yet. So yeah, unfortunately I can't give you a great answer, but that is also so another thing that one could imagine doing is trying to map with QTLs, for example, how much fitness something gained in the other environment as it adapted in its own environment, right? And it's possible that sort of this more sensitively depends on the particular genotype than adaptation in your own condition. So yeah, so I would like to address things like that, and yeah, I don't know the answer to that right now. But thanks.
Is there much variation in pre-33 across sequence sericea strains? Was the vineyard strains peculiar?
That's a great question. So unfortunately, so there are several variants between the lab and the vineyard strain in pre-33, and I don't know which the most important one is. So you can certainly, of course, tell that there has to be some variant within that gene, but of the dozen or so variants in this gene, yeah, it's not clear which is the causal one. So yeah, so I'm not sure that I can answer that directly, but.
Okay. Do you have any ideas why this relationship is linear and the same across environments?
Right. So the linear relationship, seems to imply that these things are sort of optimizing sort of one underlying trait somehow. That they're all sort of adapting along the same axis. And why that's true, I don't know. Yeah.
This is what you see somewhat conditioned upon how many generations you ran the experiment for, because
That
at
is
some point
true.
the poor ones would catch up with the good ones. If you did it after 5,000 generations, maybe they'll get the same gain in fitness as they approach some kind of optimum.
Yeah, so the only experiment that I know that directly addresses this is the Lenski experiment, because it's the only one that's long enough. In that case, they definitely, their gain in fitness slowed dramatically at the beginning, and then entered kind of a period of basically steady, slow increase, although the rate does still decrease slowly over time. Yeah, so definitely I would expect that to be true. Yeah, no, it's possible that this is a good description of sort of initial adaptation to a new environment, and a less good description of continual adaptation in a very long-term sense. That's certainly possible.
Do you know if the segments start out with the killer virus?
That's a great question, and something that I need to test, and actually I don't know. Yeah, so the S288C does not have it, but whether RM has it, I don't know. And if RM had it, then you would expect the vast majority of the segments to have it. So yeah, that's something I need to test. Yeah.
What strain were you doing? I thought you were starting with S288C.
Sorry, the first part was W303.
And
Which-
how common is like amongst all these strains?
Yeah, it's a great question. So about a quarter of them have a killer virus.
Do you know like
wild
which
strains?
ones? Escape.
Come on,
So there's, I mean, so like, SK1 has it, and I don't know, various selection of other wild strains have been documented to have it. But yeah, I don't beyond looking through, the So Reed Wickner is one of the people who studied this system a lot. And so in some of his papers, there's tables, but there's a limitation to the ones even that people have tested, right? So in general, I don't have a great list.
Could you just like have a little sequence and just like take unmapped reads of like any strain you want to and just see?
So you can, it's double-stranded RNA and it is not reverse transcribed. So that's why it didn't show up in my sequencing initially, and why you have to reverse transcribe it if you want to.
All right, cool. Thank you, Elizabeth.